Good morning, everyone. Today I would like to talk to you about some of my PhD results that I was lucky enough to do at ETH Zurich and my fieldwork I carried out here in the Seychelles. So some of the objectives of my PhD. We were interested in seeing exactly how far seed dispersal is in this species. We expect it to be low, but it's never been empirically tested. We also wanted to look at pollen dispersal. And seed and pollen dispersal are really important for shaping the genetic structure of the species. So we wanted to look at as well, what was the natural structure of the species? How did it grow? Also, where related individuals are in space affects the fecundity or the reproductive vigor of the species. Thanks. So we wanted to look at which environmental factors affected flower and fruit production. Another thing we were looking at is the sex ratios in the young plants, as currently we only knew the ratios in the adult population. We also wanted to look at inbreeding and whether perhaps the young plants that established after degradation of the habitat were more inbred than the adult population. And I'll touch upon the impacts of habitat degradation throughout, as this affects all of these processes. So I focused my fieldwork in the last 4 substantial areas where the coco de mer grows. So 3 on praline, Vallée de Mai, Font Pépère, and Font Ferdinand. And I worked on Curious Island as well. So we wanted to find the least disturbed sites we could. So we looked for clusters of plants where you could see a few large female trees surrounded by lots and lots of younger plants. So we located 4 of these in each of the subpopulations. We also located all of the surrounding males of these clusters, covering a diameter of between 80 to 120 meters. With the help of the rangers here, who I'm really grateful to because we did lots of climbing day in, day out for months, we took leaf samples for over 1,200 trees, and I extracted the DNA from these back in the lab in Zurich. So I designed a suite of 12 microsatellite markers to look at the genetic structure of these trees. So microsatellites are neutral areas in the genome, and these are biparentally inherited, meaning for each marker, they inherit 1 copy from the mother tree and 1 copy from the father. And using these, I was able to identify the mother and father from all of these individual young plants. Then, using the distance from the offspring to the mother tree, this signified the seed dispersal distance. I look at the distance from the assigned father tree and the mother tree. This indicated the pollen flow distance. So I used these genetic profiles then as a basis for the future work. And this plot at the bottom shows the pollen dispersal distances, so the father tree and where the pollen traveled to the mother tree. What I found was, as expected, but now confirmed, very short seed dispersal distances. So the average distance seeds traveled was just 8.8 meters. And in fact, over half of all seeds just traveled less than 5 meters. So here I say realized seed dispersal distance because we were just looking at the seeds that traveled and then grew up into seedlings or juveniles or immature trees. So this resulted in the very dense clusters we saw around the mother trees. We confirmed that most of these were the offspring of the female they were below. And the seed dispersal distance was much longer, significantly longer, on the steep slopes. And in Font Ferdinand in particular, the seed dispersal distances were very long, well, longer. So seed dispersal impacts on the genetic structure. So then I looked into what this looked like. So here, this plot shows the genetic structure, or the relationships between pairs of plants in space. So on the y-axis, you can see the kinship or relatedness of parental pairs, and on the x-axis, you can see the distance. So at y equals 0 across, this means that individuals are as related as would be expected by chance. This plot shows that individuals here in Vallée de Mai, up to 40 meters, were very intensely related to each other. And this took place up to 40 meters. So what does pollen dispersal look like? How did it contribute to this structure? Here, again, we saw very short pollen dispersal distances. So over 2/3 of pollen just traveled less than 30 meters, which is very short for a tree, a palm. This corresponds with the high inbreeding levels we saw across all populations and all age cohorts, even in adults that would have established before the island was more degraded. We found the shortest seed dispersal, pollen dispersal distances on Curia's Island, which indicates that the big gaps between trees do act as really barriers to pollen dispersal. And also the fact that gecko species are absent or in very low numbers on Curios, and geckos are probably the likely pollinators. This also contributed, I think, to the pollen not being able to travel. These results show that mixed modes of pollination are likely. So we expect that most pollination took place by gecko pollinators over very short distances, but sometimes there was infrequent long-distance pollen dispersal that was by wind or flying insects. And we think that this infrequent long-distance dispersal was enough to maintain genetic connectivity over evolutionary timescales. We then wanted to look at a bit more depth into the relationships between adult plants. I'll just go through this briefly because I'm running out of time. But the plot at the top, this regression analysis, shows the relatedness of plants in space. So the blue dots are all possible pairs of adult plants, all possible male and female pairs, and the orange indicates the assigned parental pairs we found from the genetics. We can see that the slopes are significantly the same, but the dotted slope is significantly higher. And this shows that parental pairs were much more highly related than expected by chance, which indicates there was a strong mate choice for coco de mer to mate with close relatives, which is really important because it's been shown in most species that the mating between close relatives reduces offspring fitness, which is inbreeding depression. And this is especially important or happens in fragmented species. So conservationists often cross distant individuals together to increase genetic diversity. But we think that this could be a problem if this happened, if this was carried out in coco de mer. Because coco de mer evolved in this way, mating between close relatives, we think that this is a stable evolutionary strategy. And crossing between two distant individuals might break up favorable allelic combinations or parts of the genome that evolved to stay together. Another thing we looked at was environmental conditions that affect fecundity or the reproductive vigor of the species. So we wanted to look at the variation in flower and fruit production and what environmental factors affected these. So we used an interesting method of tracing back the past 7 years of reproductive history. So a female tree will produce an inflorescence with a number of flowers. Some of these flowers will get pollinated and develop into fruits. And the fruits will take around 7 years to reach maturity, to ripen. And each year, a new inflorescence will be produced. So we can trace back the last 7 years of reproductive history on each plant. So we located 57 females from across the island, from a range of habitat types. So from open shrubland in the north to dense forest in the south. We measured a number of ecological variables to try and see what would affect this. First of all, we saw a really striking range in the number of flowers that can be on a palm. So these range from 6 to 123. And we found that the number of flowers was limited by the nitrogen and the phosphorus availability in the soil. And there was no difference between the two habitat types. Looking at fruit number, we found that this was significantly related to the pollen availability or the distance of the female tree to the nearest male tree. We also found over half of all fruits produced were these abnormal type, and we think that these were aborted due to pollen unavailability or incompatibility. These are much more common in the degraded shrubland. We think this contribution is that the main pollinators are absent in the shrubland. So one other thing we looked at was the proportions of males and female trees. So as we've heard already, in most adult populations, they're male biased, but we don't know if more male seeds are produced or if the females die earlier. So I tried to look for a genetic basis to this, and luckily we were successful. So I used a genomic method to try and find areas in the genome that were just related to one sex or the other, and I managed to isolate 2 markers that only appeared in the male offspring. So we screened the 700 offspring to try and find the patterns in the young plants. But we found that the ratio was always not significantly different from 50-50, which shows high stability in the species, no matter what the population size was, the availability of pollen, or the age cohort. I won't go through my summary because I'm almost out of time, but just to mention that maternal provisioning of where the mother trees funnel all the nutrients down to the base of the tree, providing nutrients for their offspring, we think that this could potentially outweigh any negative effects of inbreeding. So this is a stable strategy. But how does this affect our management of the species? We recommend that seeds should be germinated as close as possible to where they fall, obviously taking into account the poaching risk, because we don't know the consequences of disrupting this natural structure. We also recommend that closed forests should be restored as a priority, because there'll be a higher chance of successful seed production, a lower chance of the seeds becoming aborted, rather than re-establishing plants over vast areas, as was done in Curious in the early 2000s. The native gecko pollinators should be protected, and an understanding of how canopy openness and habitat quality affects the movement of these species through the forest will be important. Also, future controlled pollination experiments would be really interesting to understand how the relatedness of parents play a role in healthy fruit production, to test in the field if the results of the genetic analysis stand. I'd just like to thank everyone in SIF and the other sites in Seychelles where I worked to help me do this PhD and get all of these results. Thank you.